All right, good morning. Well, I'm echoey today. Are we okay in the back sound-wise? It sounds pretty loud. Thumbs up in the back? You guys are okay up there? What about my guys up top? You guys like the bird's eye view? Good morning, way up there. Today we're going to continue talking a little bit about fats in the body, this idea of a lipoprotein. And these are going to be a complex that are going to help transport fats throughout the body. So last day, we did talk about a protein called albumin. I'm just sharing with you the requisition form for when I got blood work done recently. Redacted all my personal information, but if your doctor sends you to get blood work, this might be a form that they provide with you, and you would go and give this to the lab, and then the lab's going to take a couple vials of blood work. How many people have had blood work done recently? Not fun, okay? It's not great, but it's a great diagnostic tool, and some of you might actually consider going into this field, okay? We have, some of our grads have gone on to what's called a clinical biochemistry program, and they will actually learn how to do some of these types of tests, set up experiments to develop new ones, and just testing for a variety of different things. So in this case, albumin's actually on here. I didn't have to get my albumin levels done. There's no little X there, but we talked a bit about that on Tuesday in terms of this protein that's in your bloodstream that helps transport hydrophobic molecules, so some drugs, different hormones, and those fatty acid chains, right, coming from adipose cells to your cells for energy. Today what we're going to talk a bit about is the lipoproteins. So these are things that will get assessed in your lipid levels. Okay, so we're going to talk about what these lipoproteins are. Other things that were on my test that I left on there was glucose levels and HbA1c. We'll look at those next week, coming back to the carbohydrate metabolism section that Dr. DeMello spoke a bit about. So today what we're going to focus on is sort of these lipid levels and looking at the different types of lipoproteins. So we're going to look at their structure and their function. Okay, so they are a complex of lipids and proteins, hence the name lipoproteins. We'll see how they travel throughout your body in order to supply cells with the various components. And we'll look at this idea of good versus bad cholesterol. Okay, so going way back to the beginning of the second half of the course, we did talk about the different structures of lipids and the structure of cholesterol itself. And you're going to see that it's not just the actual cholesterol molecule that we learned, but the different types of lipoproteins. And your liver is going to be super important. That's going to help us regulate how much cholesterol is in the body, this idea of homeostasis, so ensuring that your cells have enough cholesterol for either their membranes or forming other molecules. And then we'll think a little bit about your cardiovascular health. Okay, and these are some ideas and concepts that you can share with other people. Maybe your parents have gone and gotten blood work done. Okay? All right, so when it comes to the lipoproteins, these have kind of a unique structure. They are these transport molecules that are going to be soluble in your blood. Okay, so they're floating around in your bloodstream, and they're going to be able to carry hydrophobic molecules. Okay, so we learned the structure of our triacylglycerol molecules. These are very hydrophobic. They're going to be actually on the inside of a lipoprotein. And then there's another molecule called a cholesterol ester, which is just a modified form of the cholesterol structure that we saw earlier on in the course, and we'll take a look at how that's made in a second. Okay, but both of these molecules, these are all going to be very hydrophobic, and the lipoproteins are going to carry those molecules within the center of that structure. Okay, so you kind of have to picture this in 3D. It's kind of like a ball where it's got very hydrophobics in the middle, and then there's going to be a thin monolayer, so there's only a single layer of lipids on the surface. Right? We talked about the membrane being a bilayer. For a lipoprotein, it's a monolayer. So there's only one layer of lipids. And we're going to see those phospholipids. Remember our polar head groups with our two fatty acid tails just represented in green. The polar head groups are going to be on the surface and exposing the lipoprotein, able to interact with the solvent, so your bloodstream, helping solubilize these molecules. And then you might have a little bit of cholesterol as well at the surface. Okay, but the monolayer can't carry that much cholesterol. We really do need to form what's called these cholesterol esters in the center to help transport even more cholesterol throughout the body. So we have the polar portions of the phospholipids and that hydroxyl on the cholesterol on the surface, solubilizing these lipoproteins in that aqueous exterior, while our tags and our cholesterol esters are going to be on the interior. Okay. There's also going to be a certain type of protein, which we call apoproteins. Okay, so this is a class of proteins that can be found on lipoproteins, and they will have a similar orientation with hydrophilic functional groups and amino acids on the surface. Switch over to blue. So you're going to have your polar AAs on the surface. And then if you were looking at the structure of these proteins, you would find your hydrophobic amino acids buried on the inside of these lipoproteins as well. Okay? And so that's also going to help with solubilizing your lipoproteins, but it's also going to help with targeting. So it's going to help us help the lipoprotein tell it which cells it's going to bind to. There's going to be different receptors that it's going to bind to, and that's another way we can also distinguish one lipoprotein from another. So very unique structure of these large macromolecules, very important for transporting fats throughout the body. So when we look at cholesterol, we're not going to go through the cholesterol synthesis pathway, but it is going to be really important that we make cholesterol and provide cholesterol to our various cells, because as we've seen, it can be found in your plasma membrane. It can also be used to produce different hormones or bile salts or vitamins. Okay? And if we look at the structure of cholesterol, so here's the original structure that we learned about weeks ago. Okay? So you should be able to recognize that structure. It's got lots of carbons in it, and if we compare it to acetyl-CoA, there's actually going to be 30 steps that are going to produce the overall cholesterol molecule. Okay, and we're not going to look at that pathway. Don't worry about it for now, but do appreciate that cholesterol comes from acetyl-CoA. So if we were to track every single carbon that's found in the cholesterol molecule, you could see that it would come from either the methyl group in an original acetyl-CoA or a C double bond O. Okay, and there's probably about, I think there's close to 18 acetyl-CoAs that are going to be needed to synthesize a cholesterol molecule. And because it is an anabolic pathway, we are going to need NADPH and ATP in those 30 steps. So lots of energy molecules are going to get needed. You don't have to worry about how many. Just know that it is an anabolic process, and we're going to take all of those acetyl-CoA carbons that come from either your fats or your lipids or even protein degradation. So any of your fuel molecules, once they get broken down to acetyl-CoA, we can now use it to synthesize cholesterol in the cytoplasm of the liver. If we want to convert it from that amphipathic molecule, where we had the hydrophobic rings and that hydrophobic tail, versus that little polar hydroxyl on there, what we can do is we can transfer a fatty acid chain. So here's a fatty acyl chain that's bound to CoA, so we talked a lot about that on Tuesday. And a transferase, or more specifically an acyl transferase, is going to transfer your acyl group, right? So your long carbon chain bound to the C double bond O, and we're going to transfer that onto that hydroxyl in order to form what we call a cholesterol ester, or a CE for short. Okay, and then your CoA will just get released. We'll release that thioester bond to have that free CoA released in the cytoplasm, and now it can go on and maybe bind to another acyl chain. Okay, so we have one way of modifying cholesterol so that now we have that more hydrophobic version of it, and then that's what we're going to find on the center of our lipoproteins. So we have a 30-step reaction that's going to use acetyl-CoA, lots of ATP and NADPH in order to make cholesterol. But because it's an anabolic pathway and we want to have a way of regulating it, there's actually a specific enzyme called HMG-CoA reductase. Okay, so this is one of the steps in that 30-step pathway that's going to be super important for regulation. And when we stick a phosphate on there, right, we're going to get that conformational change, and that actually can inhibit the enzyme in the pathway and block cholesterol synthesis. Okay, so phosphorylation can do that, as well as a class of drugs called statins. So I just wanted to show you the structure of some of the statins that are out there. Okay? And one of the intermediates in the pathway is called mevalonate. So mevalonate is actually the product of HMG-CoA reductase. And if we compare it to the structure of some of the statins, you can see that they're quite similar. So we still have sort of that core structure here, and then we have larger adducts or other functional groups or molecules, chemical molecules that are going to get added onto it. And depending on which substituents you have at the different positions, you're going to get a variety of different types of statins that are present. Okay, so if you're talking to your parents or you know somebody that's on a statin, you can maybe ask them if they know how it works or which one they're on, but very similar structure to one of our intermediates. So based on those structures, and if we go back to the kinetics section of this course, we actually talked a little bit about the different constants that we can use to categorize drug binding, and statins actually lead to an increase in the Km. Okay, but your Vmax is still going to be achieved in this situation. So thinking about that, if you remember any of your enzyme kinetic stuff, can you recall what type of classification this enzyme might be? Okay, let me just pull up the Mentimeter. Come on. Let's wake up. Yeah. Yeah, no problem. All right, so we got everybody on the right track. Okay, so again, all that enzyme stuff we covered in the first half. So, the kinetics themselves, the plots, all of that you don't need to review for the final exam. But I did want to bring this into play because regulation is dependent on how molecules are going to bind to our enzymes. So when we look at our drugs, right, so these are going to be competitive inhibitors. And you can kind of tell that they might be based on their structure, right? So mevalonate is going to be made by HMG-CoA reductase. So let's say this is our enzyme. So this is HMG-CoA reductase. And if we think about this blob having an active site, so remember our active sites, these are where the reactions are going to occur, we can kind of envision some non-covalent interactions of where that molecule or that intermediate is going to be binding to our enzyme. And if we compare that to our statins, they can also bind, right? They're competitive inhibitors, so they're competing for binding to that active site, but once they're stuck in that active site, they're going to not be able to react or prevent the reaction from happening. Okay, so when you're going through in your future courses and you're learning about maybe different drugs and some of the pharmacology courses, if you're learning about any of the competitive inhibitors, just come back to your biochemistry in terms of understanding how things are binding in order to inhibit our enzymes. Okay, so we have this enzyme, HMG-CoA reductase. That's going to be our regulated step in cholesterol synthesis, and that's all going to be happening in the liver in order to inhibit the amount of cholesterol that's being made. Oh my goodness. What? Hang on. All right. So we have these lipoproteins that are going to be able to carry that cholesterol throughout the body. Okay, some of them are going to be made in the liver. We're also going to see that some of them are made in the intestine. So any of those dietary lipids that you're consuming are going to get transported via lipoproteins in the bloodstream. As they're traveling throughout your body, they're going to share those fatty acids from your triacylglyceride molecules. They're going to share some of those cholesterol molecules to the tissues in order to build up our membranes or produce energy, as well as other hormones from that cholesterol. Whatever doesn't get used is going to come back to your liver. So your liver is really that processing site. It can make new lipoproteins. It's also going to take whatever's remaining and doesn't get used by the tissues. That's what we would call a lipoprotein remnant, so whatever's remaining. And it's going to recycle it and break them up, take a look and see how much cholesterol is there, and then decide whether we need to make new cholesterol. So additional cholesterol or new tags are going to get synthesized, right? So all of these are all coming from acetyl-CoA. And then that's going to make new lipoproteins. Those lipoproteins then can go back out into circulation. So when it comes to this idea of lipid levels, getting your lipid levels tested and checking to see how much you have in the body, it really comes down to how much is being made. So if we have too many fats that are being made, maybe there's going to be defects in the receptors that are going to bind to these lipoproteins that recycle them in the liver, or maybe even problems with that enzyme, that regulated enzyme in the liver that makes cholesterol. Any issues with these can really disrupt that balance. Okay, and that's where cardiovascular disease is going to arise if you have, say, too many of these lipoproteins floating around in your bloodstream and they're not getting used for energy or used by the cells. So when we look at the lipoproteins, they actually have different compositions. So depending on how many proteins are present, those apoproteins that are on the surface for targeting, they're going to be different depending on which one we're talking about. They're going to have different amounts of triacylglyceride molecules in the center, as well as how much cholesterol is in there. I guess your percentage of proteins, that's really referring to the apoproteins. So it could be what apoproteins are there or how many. That's really going to distinguish the different types of lipoproteins. So there's really sort of three main lipoproteins. Okay, so chylomicrons is going to be one of them. And this is the one that's going to get made by your small intestine. So as you are having a meal or consuming a meal. Those bile salts are going to help solubilize any fats or cholesterol that's in your diet and package them into a lipoprotein called a chylomicron. Okay, so they're going to take any of the fatty acids or any of the triglycerides you might be consuming, packing those in, and there's going to be a protein called apoprotein 48 that's going to be on its surface, and that's how we can determine how many chylomicrons are present in your body. So after a meal, if you were testing your lipoprotein levels, you would see a huge amount of chylomicrons floating around in the bloodstream, supplying your cells with those fats and any dietary cholesterol. The second type of lipoprotein is called very low density lipoprotein, or VLDL. Okay? This is one of two that the liver is going to make. So your liver is actually going to be making this when it's synthesizing its cholesterol, when it's making triglycerides and modifying the cholesterol as cholesterol esters. It's going to package them in a lipoprotein called very low density lipoprotein. And then it can now share those tags and the cholesterol with your cells for use. And this is going to have a different apoprotein called ApoB100. Okay, and you don't have to memorize which protein goes with which lipoprotein. That's not what's important. Just appreciate that they have different components. Okay, and that's what helps us identify one lipoprotein versus another. So the liver is going to send out the VLDLs. Your VLDL is going to float around in your bloodstream. It has this protein on its surface, ApoB100, and that's going to bind to lipoprotein lipase. So your cells are going to have an enzyme on their surface that are going to bind to that lipoprotein, okay, kind of grab onto it, and then the lipase can actually hydrolyze off your fatty acids, right? So we're going to release our tags, and that's going to allow our cells to use your fatty acids. Okay. And when you lose the fatty acids, your lipoprotein is going to start getting smaller. Okay, so we go from this larger VLDL, so it's a very low density lipoprotein. Then it's going to change its structure because we're losing components, and that's going to make what's called an intermediate density lipoprotein, or an IDL. And then lastly, whatever's left over, as more fatty acids are going to get released, we're going to produce an LDL molecule. Okay, and this is what's kind of known as the bad cholesterol. So if anybody's talking about a bad cholesterol level, it means that they have lots of LDL molecules that are floating around in their bloodstream. Okay, so the VLDLs have been processed. They've gotten smaller and smaller. They're now just low density lipoproteins. The density all comes from how much protein is present. Okay, this is due to how much protein is there, right? And your LDL is essentially going to be that lipoprotein. That's our bad cholesterol. So if someone says they have bad cholesterol, just know that it's not the original cholesterol structure. It's actually this larger complex, or a lipoprotein, that's found in the bloodstream. Then the third main class, right, so IDL and LDL, these are really just variations on VLDL. Okay, it's the same original molecule, it's just getting processed into smaller types of lipoproteins. Then we have a third type, HDL. HDL is actually our good cholesterol, so this is also made by the liver. And it's going to have its own enzyme called the LCAT enzyme. And what this enzyme does is it's actually going to pick up cholesterol from the body. So as the HDL gets released from the liver, it's going to travel throughout the body. It's going to pick up cholesterol. That LCAT enzyme is a cholesterol transferase, so it's going to pick up the cholesterol, transfer it into the HDL lipoprotein, and then that's going to bring it back to the liver for breakdown and recycling. Okay, so it's really going to tell the liver how much cholesterol is actually out there in circulation. So let's just look at this in terms of figures. So starting with our first category of lipoproteins, so our chylomicrons up at the top. Following your meal, you're going to consume some dietary fats and cholesterol. These are going to get packaged into our chylomicron lipoproteins, right? Remember their structure, our soluble polar groups on the surface, lots of dietary tags and cholesterol on the inside. As it travels throughout the bloodstream, we're going to release some of those fatty acids for our cells to maybe be used for energy in, say, beta oxidation. The cholesterol might get transferred to your tissues for either membranes or making other molecules. Your fatty acids could also get delivered to adipose tissue where we can repackage them as our tags. And then whatever doesn't get used from your dietary chylomicrons, these are then going to go back to the liver. They're going to bind to receptors on the liver, on the surface of liver cells, and then they're going to get internalized or endocytosed, and then recycled or repackaged into new lipoproteins. So your liver is going to look and see what's left over. It's going to decide if it needs to make more cholesterol. It's going to repackage them and create new VLDLs. Those are going to get released out into circulation. Again, we've got free fatty acids and cholesterol that's going to get released from our VLDLs by our lipases to produce IDLs. And then when they're out in circulation a little bit longer, LDLs, again, transferring some of our fatty acids to either our tissues or our adipose cells for storage as triacylglyceride molecules. And then whatever is left over goes back to the liver for recycling. So that was our second type of lipoprotein. And then our third type of lipoprotein are HDLs. They're going to be made by the liver as well. They're going to go out in circulation and they're going to pick up cholesterol. Okay, so they're going to have that LCAT enzyme is going to pick up cholesterol from the tissues, bring it back to the liver, bind to HDL receptors, and now the liver knows how much cholesterol the whole body needs or has in order to determine whether we need to make new cholesterol. Okay, so sometimes you might hear this idea of reverse cholesterol transport. That's what HDL is doing. It's bringing the cholesterol back to the liver to show how much is actually in the body, and that can help with the regulation at HMG-CoA reductase. So here's just a little cartoon of what's going on in the bloodstream. So here's your blood vessels in here. We've got our lipoprotein lipase that's going to interact with our apoproteins at the surface of our lipoproteins, and then that's going to release our free fatty acids as well as glycerol, and then those can actually get used, travel into the tissue and get used for energy or other roles inside of the cell. Okay, so those membrane-bound enzymes are really going to help release the fatty acids, and that's what's producing those remnants or what's remaining. All right, so here's our molecule of cholesterol. So that's just our original one. Okay, and so we do get some cholesterol from our diet, and then I've also mentioned that cholesterol is made by the liver. I just want to do a show of hands. So how many people think, so we have two ways of cholesterol being found in your body, either being made or consumed, and they both contribute to this idea of having high cholesterol. Okay, so thinking about the two, do you think we get more cholesterol from our diet or your liver produces more cholesterol? So say, I don't know, over a span of a week, if we were to kind of look and see how much cholesterol are you consuming versus how much your liver is making, how many people think that diet is going to be the big way of cholesterol affecting your cholesterol levels? Okay, a couple of hands. You know, we hear a lot of stuff in the media in terms of consuming eggs and how many eggs you're going to consume. And if you eat too many eggs, that's going to affect your cholesterol levels. My mom says this all the time. Okay, and then how many people think that your liver is going to be responsible for cholesterol levels? Okay, that's true. Okay, so it is actually your liver that is going to be making the majority of cholesterol in your body. Diet still plays a role, right? We're still getting cholesterol in your diet, and I'm not saying that it's not important. It definitely is, because there's going to be other molecules that you're consuming that can affect it as well. But really, your liver is going to be primarily responsible for the majority of the cholesterol and figuring out how much cholesterol is actually present in the body. So let's just do another question. In terms of sort of this idea of regulating cholesterol levels, okay, there's actually a biochemical logic behind how exercise can lead to the inhibition of fat synthesis via kinases. Okay, so take a minute, maybe with your partner, and think about all the pathways that we've talked a bit about. Think about their regulation and see if you can come up with the link between exercise, what happens when we're exercising, and how can that lead to the inhibition of fat synthesis. All right, so we've got a couple votes coming in. So the majority are going with C. Okay, so option A, there will be no ATP for the kinases to use. Okay. Option B, glycolysis will be inhibited and no acetyl-CoA will be available for synthesis. Okay. Majority are picking epinephrine signaling will lead to the phosphorylation and inactivation of the regulated enzymes. Okay. So that's going to be our correct answer. And then D, catabolic pathways will be active to turn off anabolic pathways. Okay, so that's also incorrect. Okay, so let's just think about these ideas to begin with. Okay, so if you're exercising, we need to be generating energy, right? Your muscle cells for contraction are going to be requiring lots of ATP for that muscle contraction, right? So there is going to be ATP. You're never going to have zero ATP in a cell, okay? If you have zero ATP in a cell, that cell's dead. We're gonna see cell death, okay? So we are going to be sacrificing some ATP, right? So your kinases are going to take an ATP, and they're going to bind it to an enzyme in order to change its structure, and that's going to turn off the enzyme's activity. All right, so option B was glycolysis will be inhibited and no acetyl-CoA will be available for synthesis. Okay, but if you're exercising, you're also going to be breaking down glucose, right? So we're going to be using all of our molecules, so our glucose is going to get broken down to make ATP. We're going to be using our fatty acids to make ATP, right, in beta oxidation. So our catabolic pathways are going to be on, right? We need to make energy in our catabolic pathways. All right, so epinephrine is going to lead to the phosphorylation, right? So that was our correct answer. Just going to put this up on all the slides. All right, so we have our epinephrine signaling pathway, right? That's going to bind to our GPCRs. And then our signaling pathway is going to activate adenyl cyclase. That's going to make cyclic AMP. Cyclic AMP is going to bind to our pKa, and then pKa can do the phosphorylation in order to turn off our enzymes that are important for fat synthesis. All right, so when we're thinking about what these enzymes are, does anybody remember what enzymes we're talking about? What enzymes do we need to turn off for fat synthesis? There's two of them. Not fatty acid synthase. ACC, right? Acetyl-CoA carboxylase. So ACC was the one we saw on Tuesday. That's the one that takes your acetyl-CoA and converts it to malonyl-CoA, so that was our committed step in fat synthesis. What other fat are we talking about as well? What other things getting synthesized? Or what other lipid molecule? Maybe I'll call it a lipid. It's not a fatty acid chain, but it's another lipid we talked about. What did we talk about earlier in today's lecture? Yeah? HMG-CoA reductase, right? That's another one. So HMG-CoA reductase is important for cholesterol synthesis. Reductase. All right, so when we say fat synthesis, we're talking about our acyl chains and cholesterol as well. It's also cholesterol. Right, so there's different types of fat molecules or lipid molecules that we learned about. So there's two regulated enzymes that can get inhibited by phosphorylation. There's protein kinase A. That was the one that we saw in the epinephrine signaling pathway. There's another kinase that we mentioned on Tuesday. Does anybody remember what the other kinase is that can phosphorylate ACC? Yeah? AMP-dependent kinase. Okay, so this could be that could be protein kinase A. It could also be AMP-dependent kinase. Why does that make sense? Makes sense because if you're exercising, right, when you're exercising, all your ATP is getting used up to make ADP. And if you're not recycling that ATP fast enough, your ATP synthase can't keep up, maybe we're going to break down our ADP into AMP, right? So your AMP levels might start to rise when you're exercising. You're in this huge catabolic state. You're using up lots of energy. So that's going to help regulate our anabolic pathways or our synthetic pathways, turning them off or inhibiting them via those kinases. Questions? Does anybody need clarification, or what thing is still fuzzy about this idea? Does it make sense? Okay, so start using biochemistry to kind of understand what's going on at the cellular level. This is a great example of how everything's kind of integrating and coming together. This is how you need to be studying for the final exam. Okay, you can't think about these pathways in isolation. You have to think about everything that's going on in the cell depending on the state. Okay. All right, so talking a bit about cardiovascular disease. So, you know, when you're thinking about your overall health, right, a lot of the strategies are you should be exercising, right? You should be eating healthy, whatever that means, right? Cholesterol can contribute to that, right? But it's really your liver that's going to synthesize the most cholesterol in a day, right? So you can take a dietary approach, definitely important. We can make sure that your liver is in good functioning health, I guess you could say, because it's really going to be important for monitoring cholesterol levels, right? Those remnants are coming back. Those liver cells, one of their jobs, we're going to see a lot more jobs that your liver does next week, but it's going to be really important for looking at how much cholesterol is out in the body, recycling them, and then determining whether or not you're going to be synthesizing new cholesterol and sending it back out into circulation. So if you're consuming too much cholesterol in your diet, maybe your liver is overproducing too much cholesterol, right? Those regulation pathways might be off. We might not be turning off HMG-CoA reductase. Or if there's a problem with those receptors that are important for that lipid turnover, all of these things can help contribute to cardiovascular disease. Okay, and so when it comes to thinking about your blood vessels and what's going on, here's just an example of a blood vessel with the buildup of plaque. So when we're talking about cardiovascular disease, you might hear about this yellow, thick, waxy plaque that's starting to build up in your blood vessels. This is all originally coming from oxidized LDL. Okay, so that LDL that's floating around in your bloodstream, if it's not getting recycled by the liver properly, it can actually be oxidized. There can be chemical reactions to that overall lipoprotein structure, and as it's floating around in your bloodstream, if there are parts of your blood vessels that might be damaged, or sometimes it happens at sort of these branch points where there's lots of particles flowing through in the bloodstream really quickly, there might be areas or regions where they kind of get trapped and stuck. Okay, and we have lots of hydrophobic molecules in that lipoprotein that can't get broken down. Okay, so this buildup of plaque, that's really going to be oxidized LDL. There's going to be inflammation. There's going to be other inflammatory molecules that are going to come in and try to get rid of that lipoprotein, but it's very hydrophobic, and there's no way of breaking down any of that cholesterol or triglyceride molecules that are present. Okay, so that's really going to be what's accumulating that plaque in your blood vessels. Okay, and then that can lead to blockages, and that's when your heart won't have enough blood and oxygen, as we saw, really important for energy production, or stroke if we're talking about blood vessels that are supplying the brain. The other thing to keep in mind is that there's going to be other types of fats that can play a role. So those triglycerides, right? Again, very hydrophobic, as well as the omegas, which we'll talk about in a second. Okay, so when it comes to sort of improving your cardiovascular health or other people, really genetics plays a big role, right? It's your liver that's going to be making most of that, most of those lipoproteins, so things like that VLDLs that get sent out, as well as your HDLs. Right, so how much VLDL and how much HDL is going to be made? So we're really making HDL and VLDLs. VLDLs. And sorry, gentlemen, but women usually have higher HDL levels. That's again from the level of gene expression and the synthesis. So you got to work just a little bit harder, right? Things like exercise, as we've already seen, that can help turn off cholesterol synthesis through that phosphorylation and inhibition of HMG-CoA reductase, and as well, proper diet can also influence how much HDL is being made, as well as lowering your LDL levels. Okay. So in terms of fat intake, right, decreasing those trans and the saturated fats, but also caloric intake, right? So how many calories you're consuming all determines how much acetyl-CoA is going to get made, right? So any food that you consume is going to produce acetyl-CoA. Hopefully we can use that acetyl-CoA for energy in your cells, but if there's too much of it, that's when it's going to start being used for the synthesis of fatty acid chains and cholesterol. Smoking is discussed quite a bit in terms of cardiovascular health, and that plays a role in sort of those damaged sites that we're seeing in the blood vessels, as well as stress, right? So all of these things can help lower your blood pressure and prevent the damage, which is where that oxidized LDL accumulates. And then as we saw previously, those statin drugs can also help lower cholesterol, right? So if somebody's on a statin and they go and they get their blood work done, but their cholesterol levels are still quite high, right? Maybe they're not exercising, they're not eating healthy, they're still smoking, or they still have high stress. All of these other factors are still going to play a role. So you can't just medicate and use a drug. You need to understand everything that's going on in the body in terms of improving cardiovascular health. All right, I just wanted to bring it back to this. Do you remember what omega-3s are? I hear some chatting up here. What's going on over here? Does anybody want to offer up what an omega-3 is? Do you remember that? Have you started reviewing for the final? I really hope you have. We don't have much time. Okay. Omega-3, fatty acids. Remember, different structures. Okay. Yeah. Yep, so there's cis and trans fatty acids, and our omega-3s are going to have a cis double bond. What does omega-3 mean? Yeah, okay. 3 carbons from the end, right? We start at our methyl end, our CH3 end, and we count in. That's going to tell you where that first double bond is. In this case, these are going to be cis double bonds, help disrupt the packing of that structure, also important for signaling. And there's lots of different signaling pathways that we haven't really talked about, but these lipids can bind to different proteins, regulate gene expression, and this can all help with cardiovascular level health at different levels. Okay, so thinking about at the cellular level, so in terms of sort of what enzymes are going to get regulated, or even the DNA level. So that's really gene expression, right? Turning on and off different proteins that can help with regulating lipid levels in the body. Okay, so things like reducing the amount of VLDLs that are made, right? So that makes sense. If your liver is not sending out lots of lipoproteins, then it's not going to turn into an LDL that can get oxidized and damage your blood vessels. If we think about that reduced availability of your fatty acid chains, so if we minimize the amount of tags that are being made, then that can also influence how much VLDL is going to be produced. And so what we might instead be using is using them for your membranes. So let's use our fatty acids for phospholipids instead of triacylglyceride molecules. Use them for the phospholipids that our cells are going to need. Build up those cell membranes instead. Okay? Or we can use them for energy, right? So we can use them as an energy source, supplying the cells with ATP. Okay? And if we're thinking about what enzymes might be regulated at the level of gene expression, we can reduce the amount of ACC and FAS that are actually made, as well as their activity regulating them in the liver as well. Okay, so again, you don't need to know all the details, just an older review article, but I kind of liked it, the way it's representing the different tissues throughout the body and what's going on in terms of thinking about how we can regulate fatty acid levels. All right, so key points, go back, make sure you understand cholesterol is made from acetyl-CoA. We have our regulated step, HMG-CoA reductase, and it's these lipoproteins that are going to be really important for traveling throughout the body and determining whether you have good or bad cholesterol in your body. Don't forget to start working on your final assignment. Keep up with your review, please, 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 and we will see you next week.